Hello students, we are here with one of the most important topic that is the gram staining and which is going to be the important one for your XC and Excel exam in your food technology background. Okay, so today we will see this topic. What is gram staining? What kind of questions are going to be asked in the exam from this particular topic that is known as the gram staining. Basically, this topic is from your food microbiology in your food technology section, and this food technology section is common in both the exams, that is in get XC exam and get in Excel exam. Okay, so now let's see the topic. So our today's topic is gram staining. I hope that you all may be aware about the gram staining, okay? Because this word is, we have heard it for the search several times, and it is the common term. So basically, in microbiology, if you people have the background from the microbiology, little bit of background, then you may have heard about the gram staining, okay? So this is the kind of a staining technique, and this staining technique is discovered by the scientist known as the Christian Gram, okay? Hence the name gram staining is given over here, okay? So, firstly, we should know, like in food microbiology, why the staining technique is given. Okay, what is the importance of the gram staining? And before going towards the gram staining, you all should be aware about the microbiology. So the thing is, when you go for the food microbiology, so it has the study of the microorganism, basically the microorganism that will get colonized. Here you can see, then they will get modified, and the process or the contamination of the food spoilage. So all these aspects together are considered under the terminology or under the term that is known as the food microbiology. So microorganisms which are used to transform the raw food into the gastronomic delights, or into the product, fermented product like your cheese, pickles, or that can be your soya sauce, or else you may have like you people know about the wines and the beers, all the alcoholic beverages. Basically, they are gone through the process known as the fermentation. So these all aspects are covered under the food microbiology. On the other hand, your microorganism, they can degrade the food quality. Okay, they can degrade the food quality, and thus they can lead to the spoilage. So this spoilage section is also considered under the food microbiology. So what is the role of food microbiology? So to detect, okay, detection and the control of the pathogen, to detect the pathogen and then to control them. So that is the most important role of your food microbiology. Okay, and this is the important part. Now we will see what is the gram staining. So basically, gram staining, as I told you, it is discovered by the scientist, the whose name is basically Christian Gram. Now the thing is, like this is the kind of a staining technique. Now, what are the dyes and what is the staining? So for any type of staining, staining means you have to impart a particular color to your sample which you have prepared, a microbial sample or bacterial sample. So the thing is, many types of dyes can be used. Okay, you can use acidic dyes, or either you can go for the basic dyes. So that depends. So the many type of dyes are used to stain the microorganism. Now they have the two features in common. Like dyes have the two feature in the common. The one feature is they have the chromophore group. Okay, and the second feature is they have the conjugated double bond. Now these both the features are there for the dyes. So when we go for the dyes, you will have the basic and the acidic. Now the thing is, according to your exam point of view, what you have to remember, you should know the name of the acidic and the basic dyes. Okay, that is what the most important point over here is to remember the name of the acidic and basic dye. Thus, you have to remember which name or which type of dye is going to be used in the procedure of the gram staining. Okay, and besides this gram staining technique, there are various other type of staining techniques. Now, when we go for the dyes, so in types of dye, you have the acidic dye, you have the basic dye. Now, the basic dyes example here are enlisted. Basically, you will have the methylene blue, basic fusion, crystal violet, saffron, malachite green. So these all are the example of your basic dye. Now this basic dye will have the positively charged group, and they have the generally they are sold as the chloride salt. Okay, so these basic dyes bind to the negatively charged molecule like the nucleic acid, many protein, and the surfaces of the prokaryotic cell. These basic dye are going to be bind to the nucleic acid, the protein, and the surfaces of the prokaryotic cell. When you go for the acidic dye, in acidic dye, you will get the eosine, rose bengal, acid fusion. So these all are your nothing but the acidic dye. Now the charge group are present in the acidic dye. So the group like the carboxyl group, COOH. So this is nothing but carboxylic acid group, acid. Okay, so from the acid term, you can remember this group is present in your acidic dyes. Now there should be some trick and tips now to remember the name of the basic dye or acidic. Like suppose if you go for the acidic dye, there are only 2 to 3 names: eosine, rose bengal, acid fusion, that you can work up. Okay, that is the another thing. But if when you go for the basic dyes, there are many names, and to remember them is a little bit of tricky. So here I have given one trick to remember the names of the basic dye. So that trick is basically you can remember it as MMBS, or either you can remember as MBBS. Okay, it's up to you. So MMBS का CV basic है. Now from this, how you can elaborate the name of the basic dye. Now this M will stand for your methylene. This M will stand for your malachite green. Okay, malachite green. Now this second M, this will stand for the methylene blue. Now this S here will, sorry, this B here will stand for the basic fusion. Or else you can do it like this: methylene blue, MB. Okay. First M, malachite green; MB, methylene blue; S will be saffron. Okay, here S will be saffron. Now after this cell S, you have the C and V capital. Now CV is nothing but your crystal violet. C for crystal, V for violet. And basic A. Now this basic is nothing but basic fusion. So this is how you can remember name of the basic dyes. And acidic dyes are only 2, 3 in number, so that you can remember easily by just mugging up. Okay. I hope this is clear. Dyes and staining. Now the important point come over here. That is the gram staining. But before going towards the gram staining, what you should know, you guys should be aware about the difference between your gram positive bacteria and gram negative bacteria. Because why we are differentiating them, why we are studying them. So there must be some difference. Now, this gram staining is nothing but it is kind of your differential staining technique. Now, when you go for the study of the differential staining, unless and until your bacteria will have the some kind of difference between them, so if they do not have any kind of difference, then how you will get the staining, how you can apply the staining techniques, or even after applying the technique you will not see any distinct difference. So firstly you should understand the difference between your gram positive and gram negative. Okay, that is the important thing. Now, when you have to understand the difference between the gram positive and gram negative, this is the basic term. Probably you guys may be aware about this already, but still we are repeating over here. That is the difference between the gram positive and gram negative. When you go for the gram positive bacteria will have the thick cell wall of peptidoglycan. Okay, their cell wall is made up of the peptidoglycan. That is the outer membrane is absent in them. While in the case of gram negative, there is the thin cell wall. Okay, you should remember this. Gram positive, thick cell wall; gram negative, thin cell wall. Outer membrane is present in the case of gram positive. Outer membrane is absent in the case of gram negative. Now, when you go for the teichoic and lipoteichoic acid, so that will be present in positive, it will be absent in negative. When you go for the lipopolysaccharide, so lipopolysaccharide that is absent basically in gram positive, but it is present in gram negative. And when you go for the bronze protein, so that bronze protein is absent in gram positive, it will be present in your gram negative. Now the main point, like after applying the overall staining technique, after doing the whole procedure of the gram staining, which color you will get? So your gram positive bacteria, it will stain purple in color. Okay, gram positive bacteria will stain purple in color, while your gram negative bacteria, it will stain pink in color. Okay, remember this: gram positive will stain purple in color, gram negative it will stain pink in color. So this is the basic difference between your gram positive and the gram negative. Now, what is the differential staining? Okay, what is differential staining? What are the types of the differential staining? And like what is the gram staining? This is the sequence which we are going to study. So in differential staining, the procedure that are used to distinguish the organism based on their staining property. So that is defined as the differential staining. Okay, it is nothing but it is the kind of a procedure which will allow you to differentiate between the organism by doing the staining procedure. So if you use the gram stain, it will divide the bacteria into the gram negative and the gram positive. This is what we have seen in this slide, the difference between your gram negative, gram positive. So that will be there. This differential staining technique, it will help you to differentiate between the gram negative, gram positive. Now, what are the types of the differential staining? The gram staining, we are aware with the gram staining, okay? And we are going to learn this today. Now, besides this, you will have the acid fast staining, you will have the endospore staining, you will have the flagella staining. And all these other staining techniques are also important. But today we will study the gram staining, okay? Now, what is the procedure for gram staining? What will be your what is the procedure of gram staining, how it is done, what are the dyes used, whether acidic dyes or basic dyes are used. So all those things we are going to study in depth, okay? And all these techniques already, like besides gram staining, you will have the acid fast staining, you will have the endospore staining, you will have the flagella staining. So all these things we have covered in depth in our live classes, that is in our batch of GATE, XC and Excel, that is 2024. Okay, in that batch we have covered. And if you want to see this topic in depth, then join our batch today, and you can get the good hold on this topic. Okay, so now we will see how gram staining is being done. So in gram staining, we know what is the procedure, okay? But you guys may have the question in your mind, what is the difference between the differential staining and the simple staining? Simple staining. Now in differential staining, what you have seen is you have the, basically it will differentiate between your gram positive, gram negative, okay? Now the thing is in simple staining, your microorganism, they can be stained by the simple staining technique also, in which only single type of dye is used. In your gram staining, now you will use many types of dyes. Many dyes are used. While in case of your simple staining, only single type of dye is used. Now, the simple staining technique values, it lies in the simplicity and the ease of use. One cover the fixed smear with the stain for a short period of time, and it is going to wash off the excess stain with water. Okay, means firstly you are going to apply the dye or the smear with the stain for a short period of time. Then you are going to wash it off. After washing, whatever excess stain will be present over that your slide, it will get washed off, okay? And blot the slide dry. Make the slide dry just by using the blotting paper. Now the basic dyes like the crystal violet, methylene blue, and the carbon fusion are frequently used in the simple staining technique. And why simple staining technique is used? It is used to determine the size, the shape, and the arrangement of the prokaryotic cell. So for these type of reasons, your simple staining technique is going to be used. Okay. So just remember the one basic difference between the simple staining and the differential staining. That is the single type of dye is used in the simple staining technique, and many types of dyes are used in your uh, gram staining technique. That is nothing but in your differential staining. Okay. Now we will see your gram staining technique. See, now in gram staining, as the name suggests, the gram stain. So it is developed in 1884 by the Danish physician Christian Gram. Okay, it is developed in 1884 by the Christian Gram, and it is most widely employed staining method in bacteriology. Means even if you are going to study the microbiology, or if you are studying the food microbiology, bacteriology, whatever, whenever the microbiology and the staining comes, the first name that will be in your mind is nothing but the gram staining. Okay, you will not remember like what is the flagella staining, what is the acid fast staining. That you can remember when you are going to study them in depth. But whenever only you have to just memorize the name of the staining technique. So at that time you will remember the gram staining technique. Okay, this is most widely employed. Now this gram staining, na, it is nothing but it is your four-step procedure. Okay, it is your four-step procedure. In this four-step procedure, the first step will be your smear or the sample. The sample which you are going to take, that is known as the smear. So the smear or the sample, it is stained by using the basic dye. Now which basic dye is used over here? So the first dye which you are going to use is crystal violet, CV. Okay, crystal violet. It will penetrate and the cell become purple in color. Now both the cells, you will have the two cells. Okay, now both the cells will become purple in color by applying the crystal violet, because this crystal violet is going to apply the purple color to your sample. Now in step 2, what you are going to do? you will use the iodine as a mordant. Now what is the function of mordant? Like what is mordant, what is its function? So mordant is nothing but by using the iodine, or the function of mordant is basically it will increases the mass and size of the crystal violet dye, and thus it will form the CVI, that is nothing but your crystal violet iodine complex. Okay, CVI complex, that is the crystal violet iodine complex. So this mordant will work to increase the mass and size of the crystal violet. Okay, to increase the mass and size of the crystal violet is the function of the mordant, and that mordant here you are going to use iodine. Now in step 3, this is the step 2. In first step, what you have done, you have applied the color and your cell become purple in color. In step 2, you are going to use the crystal violet, that is the model. In step 3, you will use the decolorizer. You will decolorize the cell. Now alcohol is used as a decolorizer. Okay, alcohol. Now you will decolorize the cell by washing with the ethanol or acetone, that is alcohol. Alcohol what will do? It will dissolve the cell membrane and make hole in it. Okay, this is your bacteria, and if this you have the cell. Now after applying alcohol, what will happen? This is your bacteria. This cell membrane will get dissolved. Holes are here. Okay, holes will appear in it. So this is the function, or this is the scene which is going to happen after applying the alcohol. Now what will happen? Gram positive cell wall will remain purple. Okay, because what we have seen, we have already studied difference between gram positive, gram negative. Gram positive will have a thick cell wall. As the cell wall is thick, so there will be no effect of the alcohol on the cell wall of gram positive. But gram negative will have a thin cell wall. Now what will happen? After applying alcohol as a decolorizer, that thin cell wall, it will get breakdown, means hole will appear on that cell wall. Here in this picture, which I have drawn, here you can see like how these holes are made, okay, by using the alcohol. So this is what happening after using the decolorizer or after using the alcohol. Holes will be made, so what will happen? Cell will lose its color. It will lose its purple color. Means your gram negative in the third step, na, your gram negative will become colorless, but your gram positive, it will remain a purple color. Okay, that is what you here happening. While gram negative get decolorized, means it will lose its color. Now in fourth step, you are going to do the counter staining. Counter staining means you will apply the saffronin to both the cell. Saffronin is pink or red in color. Now what will happen? The cell which is already purple in color, it will remain purple. Okay, that is gram positive. Even if you are going to apply the saffronin as a counter stain, it will remain purple in color, because why? Saffronin will not penetrate. It will just impart the color. Now already your purple color is penetrated in your gram positive cell. So it will not be loosened, or it won't change by applying the saffronin. Okay, it will remain as it is. But in the case of gram negative, what we have seen in the third step, what you guys have seen in the third step, your gram negative cell has become the colorless. Why? Because it has the thin cell wall. So as it has become the colorless, so what will happen when you are going to apply the counter stain, that is the saffronin, that colorless cell will become pink or red in color. It will take the color of the saffronin, that is your counter stain. Okay, so at the end, what you will get? So you will get your gram positive as purple, and gram negative will become which in color? Pink. Pink due to what? It will become pink due to saffronin. Okay, so this is what the difference is. So all the this telling now, like the difference between the gram positive, gram negative is based on its structure, which we have seen, cell wall structure. Okay, and the second thing is that we are going to see, that we have seen basically is the stains which you are used, because by using the alcohol, your gram negative loses its color and hence it become colorless. And after applying the counter stain, that is the saffronin, which is purple in which is pink in color, your gram negative cell will take the pink color. Okay, so this is what happens in the gram staining technique. It is basically your 4-step procedure. Now in this figure, you will have the more clear idea. See, the first is crystal violet primary stain. Both the cell will become purple. Here you can see gram positive, gram negative. Both the cells are purple in color. Now the second step, you will apply the iodine as a mordant. Cell will remain purple in color. Okay. You people know what is the function of mordant to increase the cell size. Thus you can see this clearly. Now cell will remain purple in color. In first step also it is purple. In second step also it remain purple. In the third step, after using the decolorizer, gram positive become purple, gram negative become colorless. Why? Due to because the cell wall of the gram negative is thin while positive is thick. So thin cell wall will lose, it will lose its color more rapidly and easier to lose. Okay. Now here what you see, this is negative, this is negative. Hence it become colorless while all the positive cells are purple in color. This is the third step. In the fourth step, you are going to apply the saffron in. That will be your counter stain. When you have applied the saffron as a counter stain, your gram positive will remain purple, negative become red. We have seen these two are negative. See, these are pinkish to red. While this become purple. Now the pinkish to red is nothing but negative. This purple one is nothing but your positive. So this is what going to happen in the whole procedure of the gram staining technique. Okay. I hope this is clear to you people. What is the gram staining? Which are the dyes used? Now you have to remember the names of the dye and the whole four-step procedure. Now in next slide, I will show you like which type of questions are going to be asked from these kind of this topic, that is the gram staining. In previous year GATE, how which type of questions are asked. See, this is just the thing, like simple staining technique will give you the image like this, while in the differential staining, this pink color one are your gram negative, okay, and this purple color are nothing but your gram positive. Okay, these are gram positive, these are gram negative. Now, which type of questions are asked from such topic? Now, see, the question is the lipopolysaccharide, that is LPS, in cell wall is the characteristic of, means in cell wall means it is present in. We have seen it is present in gram negative bacteria. Where we have seen, see, here in gram negative C, LPS lipopolysaccharide is present, while in positive it is absent. So that's why I told you like how the small things have the greater impact on the questions that are going to be asked in your exam. Okay, so see, you may have in your mind like why this simple kind of things will be there in the gate exam is so tough and all this, but the thing is now they are going to check your basic knowledge. Okay, that is the most important thing, to check the basic knowledge. Hence such type of questions can also be there in your exam. So LPS in cell wall, which is a characteristic of gram negative bacteria, which we have already studied. Here the explanation is given, like what is the difference. So the thing is, explanation is nothing but that table, difference between gram positive and gram negative, that is okay. Now the second type of question that can be there is the what is the correct order of the staining reagents in the gram staining technique. See, they have asked about the reagents. So if you want to do the if you want to arrange the sequence for the reagents, now the first thing which you guys should be clear with is the procedure, the four-step procedure. Okay, unless and until you guys are not no, you guys are not clear with the procedure, you are unable to solve this question. Okay, and this is nothing but the PYQ, that is the previous year gate question. So here the correct option is in step 1, you will have the crystal violet, which will stain the purple color cell. Step 2, you will use the iodine, that will be your mordant. Step 3, you will use alcohol, that is the decolorizer. Step 4, you will use saffronin, that is the counter stain. Okay, so this is what the thing is. Okay, these type of questions will be there in your gate exam, hence this topic is very important. Okay, so now we have covered all the basics of this topic, that is your gram staining. This is the one of the most important topic from your food microbiology, so that's why it is important. All the important points are covered, and there are many more things to be know about the gate about the staining techniques. Okay, that will be there in our the live session and in our target batch. And if you guys want to crack the exam, that is the GATE XC or Excel in food technology for this year, that is year 2024. So don't wait and join our target batch, that is GATE 2024 XC and Excel now. Okay, I hope you like this video. You will like such type of information, and for getting such more information on the most important topics that will be there in your GATE exam, hit the like button and subscribe on our channel. Okay, thank you.